How about that?
Yeah.
Yeah.
Yeah. Okay. Good.
So the institute was founded in 2000. That's when I joined the institute, and its special mission is to take on complexity and use a systems approach to understand the basis of that complexity, so you can then apply it towards medicine and even education. And I'll touch upon those ideas a little bit, but I'll just jump straight into my talk. It does seem like a diverse set of topics, and while the questions are in very different domains, I think at the heart of all these questions is the notion that these are complex systems, and understanding the complexity will allow us to come up with rational ways of understanding why individuals are different, why pathogens gain resistance to drugs, how microbial communities respond to climate change in ways that are non-linear, and so forth. And to do that, you really need to move away from a reductionist approach where you look at individual organisms or genes and take a look at the system as a whole. And the approach to do that is what we call a systems biology approach. And the objective here is to build a predictive understanding of these complex behaviors, so you can then use rational means to manipulate that behavior. So at the heart of this approach is predictive biology, and I'll spend some time on telling you what I think of in terms of predictive biology. And as I go through some vignettes of how we apply it, it will become clear that the way we are practicing science is very different from how we teach it. So I'll, with time permitting, end with this idea of teaching science the way it's practiced, and how does one go about doing that. And so predictive biology, the idea that you can predict biological behavior, now it's accepted, it's been seen, but there's fundamental questions underlying how you do it and why you can do it. Now, how can you take a complex system that has thousands of genes, trillions of cells, hundreds of cell types, if you take the human system, and how do you predict its behavior? How is it even possible to do that? So there are some basic underpinnings, evolutionary underpinnings of why we are able to do that. And the underpinnings have to do with the way the environment is structured. The environment is not random. You have changes in the environment that are coupled for physical chemical reasons. So you see a change in one factor, and the consequence of that change through chemical interactions and downstream consequences propagates across other changes. So there's covariance in different environmental factors. Now, from a perspective of an organism, they see these changes repetitively through their evolutionary history, and as a result they can take advantage of it. So they can see a change in one factor and exploit it by preparing in advance to a future change, and maybe preparing resources and enzymes to metabolize some new resource that could come about in its environment. And you can actually observe this happening in the laboratory. So what we did a few years ago was we took yeast and asked, could we teach yeast some new tricks? Can we teach yeast to sense some new unnatural environmental factor from its evolutionary perspective, so it can then get cued for a future lethal event? So we put yeast in this artificial environment where we exposed it to caffeine, and then after 30 minutes we exposed it to this toxin called 5-fluorouric acid, and we did this every 10 generations or so, and every 50 generations we asked, now if you take this yeast population that is evolving in this novel structured environment, now if you give it caffeine, is it able to remember what it's seen in the past and then elicit a preparatory response, adaptive response to the upcoming lethal challenge? And so we demonstrated that within a matter of 200 generations or less, yeast was able to gain this, internalize this information, and next time it saw caffeine, it was able to elicit this protective response in advance. And we know it is anticipatory because if you remove that time difference of 30 minutes, you don't see the advantage of seeing caffeine. So there's a time dependence and causality in there. And we went further to ask whether we can find out how it had done it. So we set up an experiment where we gave yeast the opportunity to gain this protective response by rewiring its networks. And so there's an enzyme that is required for making a nucleotide called uracil, and that's the enzyme that gets poisoned when you add 5-fluorouric acid. And one way to get protective response would be if that enzyme function is somehow masked or killed when you have 5-fluorouric acid. So we gave yeast this opportunity that when it saw caffeine, it could translocate this enzyme into this organelle called peroxisomes and get protective response. But when the toxin goes away, the enzyme has to come out to make this nucleotide so DNA can replicate. And we showed that the two networks, the one for sensing caffeine, within 50 generations got rewired to the translocation network, which was completely unrelated. So when now yeast saw caffeine, it was able to translocate this enzyme into peroxisomes, and then the enzyme came out when the toxin pressure was taken away. So what this tells us that the networks within organisms are highly flexible in terms of how they learn from the environmental exposure. So they can internalize information and use that information for getting a competitive edge among the other organisms in the environment. And so the structure of the environment is mirrored in the structure of the way processes are connected inside a cell. So you may have changes in temperature and salt solubility and so on the outside. Inside the cell, the way the organism deals with heat stress, salinity stress are coupled in a way that reflects the external coupling. Now there's downstream consequences of this. One is if the environment continues to be structured, the organism benefits from it. But if you decouple the environment, which was shown by Saeed Tavazoi, who's at I think Princeton or Columbia now, if you decouple the natural coupled environmental changes, the fitness of the organism drops. Okay, and there's another consequence as well, which is although they can potentially adopt infinite numbers of states, because of the structured environment, the organisms adopt few dominant states, and because of this we can begin to learn how they're able to transition across this limited number of states. And one way to do that, and what we demonstrated, was you can take any organism, you can perturb it in some meaningful way, that is ecologically meaningful, that is, and then observe how it responds. And you can do profiling of all the changes in gene expression, you can do phenotypic measurements and so forth, and then you can reverse engineer the topology of interactions amongst all components of the cell, all the genes, proteins, RNAs and so forth, to then explain how the incident changes were processed into the outputs that you observed. And furthermore, you can predict responses to new behaviors. And there are two key ways to do this. The first step is you take these large amounts of data that come from these perturbation experiments, and then you ask how are all the genes organized in a modular fashion such that given the environmental context, the right set of genes come together and behave coherently to deal with that environmental challenge. And that's called conditional co-regulation. And this is an algorithm, it's a biclustering algorithm. It essentially bootstraps through all the gene expression data sets and looks for genes. So what you have here are different environmental conditions. It could be different patients, it could be different kinds of drug treatments, for example, and these are relative gene expression changes, and the algorithm can bootstrap its way through this large data space and find genes that are co-expressed in subsets of patients or environments. And the discovery process can be semi-supervised by requiring that these genes also share some common regulatory mechanisms, because you don't want to get misled by coincidental correlations. So you want to find genes that are co-regulated, not just coincidentally co-expressed. So you can look for evidence in their DNA that there might be a protein that is binding and mediating this co-regulation that you observe, and you can look at possibly interactions among different proteins and use that as a mechanism to identify which genes to group into each cluster. And you can do this discovery thousands of clusters at a time simultaneously. And so you can organize this very large set of data, multi-omics profiling data sets, into this modular sets of conditionally co-regulated genes. Now the question is, what's the wiring diagram that is responsible for mediating these co-expressed or co-regulated changes? There's a variety of ways you can do it, and one is you can look for these mechanisms de novo, as I described. The second thing you can do is you can look for binding sites that are shared across the genes. And if you're not familiar with transcriptional regulation, one way that genes get co-regulated is by a protein that is binding to a control region in front of the gene, and that protein then turns on all the genes in front of which it is bound. The question is, where in the genome is it bound and active? And in humans, there's thousands of transcription factors, and each transcription factor can recognize a particular region in the DNA by recognizing maybe 6 nucleotides or so. So you can look at where these sequences are distributed across the genome, and then you can bring in other types of information. There's this information called encode profiling, which tells you whether the chromosome is accessible or not. So in this example, there's 3 possible locations this transcription factor can bind. Only 2 of the locations were accessible, meaning the chromatin was open in one or few of 200 cell types that were studied by this ENCODE consortium. And you can zoom into these 2 regions and find that of the 2, there was only one that had evidence of a protein actually being bound and protecting the DNA from being cut by this profiling technique. And you can come the other way and confirm the prediction here is of the 3 locations, only 1 location is actively bound by this transcription factor. So we can come in and from the other way and say, if we were to take an antibody and pull out this protein along with the regions it's bound to across the genome, would we find evidence that it is bound in this location? And we see that of the 3 locations, there's evidence for only 1 of the 3 locations. So we've done this for all the thousands of human transcription factors for which these recognition sequences are known, and we have this big map, and we can now look into each module and ask, are these active sites enriched within these modules? And so we can draw a wiring connection from the transcription factor to that module. This is one way to do it. We've done that for microRNAs, which are small RNA molecules that control gene expression. The other way you can also do it is, and this works for prokaryotic systems, so these are bacteria and archaea. You can do a regression based approach where you can say we can try and explain the average expression change of genes within a module as some weighted sum of weighted changes in transcription factors. Okay, and by doing so you can then derive models like this where you can say there are 2 transcription factors or proteins that act together with these 2 environmental factors to control genes in this module. And this type of a model can be used to predict gene expression changes, and we've shown that you can predict very accurately how genes within a module are controlled by these regulators, even in new experimental environmental conditions, as is shown here. And not just for one module, but you can do it across the entire genome. And we've done this for prokaryotes, we've done this for eukaryotes, for chlorophytes, and I'll give you 3 vignettes on how this can be used. But for eukaryotic systems, for humans especially, we've also built this pipeline based on similar principles, but remember the biology is different from bacteria to archaea to humans. So taking that into account, as I showed you with the transcription factor wiring map that we can do with chromatin accessibility, we built a lot of these databases, and now we can take large amounts of data from efforts such as the Cancer Genome Atlas, where there's over 30 cancers that have been profiled across tens of thousands of patients, and we can bring that information in, and through sequential steps finding the co-regulated modules, those that are associated with disease, and those that have got mutations driving the changes that are observed across patients, we can build maps like this, where we can say that there are these mutations, subsets of mutations across a cohort of patients that we have evidence that they causally alter the control mechanisms to have downstream consequences on these modules that are associated with the disease, such that we can then rationally identify drugs individually or in combination. And this is one way to personalize therapy to patient characteristics, and I'll get to that later on. And because we've built this conceptual framework that is generalizable, any advancements we make on any of these problems that I'm going to tell you about has impact on making advancements in applications to the other domains where we're focusing on. So from microbes to host pathogen interactions to cancers, all the way to microbial ecology and ocean microbiomes and so on. So let me jump into 3 vignettes and tell you how one can apply this foundational approach to understand some very complicated problems that have to do with climate change, have to do with infectious disease, and I'll talk about tuberculosis, and then I'll end with personalized medicine and cancers. So one of the biggest things we're trying to do in my lab is predict when microbial communities might collapse. Okay? And this is of high relevance given how much we abuse the environment, such as in agriculture, we've used so much fertilizer, for example, and increased nitrate levels that has resulted in disruption of the microbial interactions and loss of gigatons of carbon. And if you look inside the soil, you have very complicated sets of interactions that exist among different groups of microbes, and they process carbon either by respiring oxygen. If you go lower in depth, they might respire nitrate, sulfate, metals, and so forth. And then as you go further below, there's all kinds of interactions that are important to drive this process. So when you add something like nitrate in excess, you really shift the equilibrium of these interactions. And as a result, most plants that we have in terms of food production don't have the natural microbiomes. And we're working with a large group of scientists across all these institutions in a project called Enigma, where we're looking at a Superfund site in Oak Ridge, where we're trying to understand the consequence of the high radionuclide and nitrate contamination. It's a Superfund site, and it's beginning to leach into groundwater. And we're trying to understand how that contamination has shifted the microbial community dynamics, and whether we can predict what would happen when there's a catastrophic event. Say whether when there's a rainfall, if there's nitrous oxide emissions, where would it happen, and what are the organisms responsible for that, and so forth. So to do this, we have a large group that's working in the fields, and they're taking measurements of changes that are happening across the field site with enriching microbes, and then we have connections all the way to the laboratory. And I'm working across that transect, but I'll tell you a story on the laboratory side. It's a story that came about through collaborations with David Stahl in the civil engineering department here. And he set up this synthetic community of two organisms, a sulfate-reducing bacterium and a methanogen. Now, the sulfate-reducing bacterium can break down compounds like lactate by using sulfate to respire. But when sulfate is missing, it then interacts with this organism, a group of organisms called methanogenic archaea, so it can drive the reaction forward. Otherwise, you have accumulation of hydrogen formate, which inhibits this breakdown of these carbon compounds. So the question was, if you put these two organisms together in the laboratory, how do these organisms evolve in a way that they start taking advantage of each other? So we can understand how structure and function emerges in microbial communities. And then we can begin to understand in what context they might collapse. And just to give you a sense of how this experiment was done, very quickly, so we took these two organisms, we combined them, we set up 12 lines, meaning 12 tubes that were shaking, 12 tubes that were not shaking, and then tracked over 1,000 generations how they evolved by looking at all kinds of phenotypes and also by sequencing the communities down to getting clonal isolates of evolved communities and pairing them and looking at how they might complement each other. We even went down to single cell level sequencing to understand how evolution might have happened in across the two organisms to drive coevolution. And then we've done experiments to look at how the community might be able to adapt to changing environments and whether we can predict collapse. So just to give a sense of, this is a very data rich set of experiments, and from this we started mining information regarding how these two organisms came about to love each other. And what we observed was that within 300 generations, the growth characteristics of the community improved dramatically. And then we went in and sequenced the genomes of the evolved communities and saw some interesting trends. One thing we saw was across all the lines, there were certain sets of genes and pathways that were mutated, albeit in different locations. It almost seemed as if there was convergent evolution, the strong evidence of convergent evolution. And so what you're looking at here are, each panel here is a gene, and across x-axis is the coordinates on the gene, and then what you have here are, across x-axis are the different evolution lines, and across the y-axis are the coordinates of the gene. And you're essentially looking at how the gene was mutated across all the lines in different locations. And the mutations have moderate to severe effects. based on the kind of change it was. And so these are genes involved across a set of pathways that seem to be essential for the collaboration between the two organisms. And most notably what we saw was in the absence of sulfate respiration, because we put them in a condition where sulfate was not present, so they depended on each other. The pathway for sulfate respiration was almost deterministically lost across all the evolution lines. And then to confirm that there was coevolution, we then took an evolved community and we teased it apart, and we got individuals of the community, and then we started pairing them. So when we look at the evolved community, we see that the fitness of the community has increased dramatically compared to the ancestral community. Okay, so if you take the ancestral pairs, they have low growth rate. Now if you take any individual evolved member, so if you take the evolved Methanococcus or the evolved Desulfovibrio, you have improvements in fitness, but when you combine both evolved pairs, you have this dramatic synergistic improvement in fitness. To understand that, as to how that might have happened, we did single cell sequencing and figured out the lineage of mutations. So what you can do is take a population of evolved prokaryotic cells, do single cell sequencing, look at the frequency and distribution of mutations, and based on that create this hierarchical tree that explains what sequence the mutations might have emerged. And then we can go back to our ancestral mutation sequencing, because we tracked the populations at different times and then cross validate to confirm that the lineage was accurately inferred. So we can, each node here is a mutation, and you can see the sequence of mutations that happened, and we see there are some very specific things that happened as the communities evolved. And there are two lines here. This is the sulfate respiration negative, and this is the sulfate respiration positive. The reason I'm showing you this is when this event happened where sulfate respiration was lost, there was this radiating effect where mutations started accumulating across the information processing pathways, the regulatory networks of this organism. And we thought this might be because sulfate respiration is not needed in this obligate kind of uniform resource environment. So you can get rid of the regulatory functions, because regulation is required when the environment changes. If the environment is uniform, you don't need regulation. So our hypothesis was, because this obligate relationship existed in a uniform environment, like any organism, unnecessary pathways would be shed through evolutionary processes, which would mean that if we were to take the organism, the community, and shift it from requiring sulfate to depending on each other, regulation would be important, because you would have to turn on genes required for one function and then the next, and back and forth. And so we set up an experiment like that, and then we apply this network inference approach to figure out what are the regulators of this process, so we could mutate it. And then the hypothesis would be the mutant that can't regulate would not be able to survive in this fluctuating environment, whereas the wild type would fluctuate quite effectively. And to our surprise, we saw the exact opposite thing. We saw the wild type almost deterministically across 24 different lines went to collapse, whereas the mutant, there were a couple of mutants we had, it indefinitely continued to transition between syntrophy and syntrophy and sulfate respiration, syntrophy and sulfate respiration. And then we couldn't explain this initially, and then we realized that the regulation that was happening in the fluctuating environment became too burdensome for the organism to continue fluctuating between one state to another. So we looked inside the cells to see, could we tell, could we have predicted this? Could we have predicted when the systems collapse? So we sequenced the transcriptomes at each of these points to see if there was any signature of collapse that emerged. And what we observed was quite interesting. What we saw was, in the wild type, the internal state, remember I said the external structured state of the environment is mirrored in the internal state. So you see in the wild type, when it's growing in syntrophic conditions, the internal state looks like it's a syntrophic state. Then you move to sulfate respiration, it shifts, but as you transition, it goes farther and farther away from the desired state. So eventually the internal state is completely decoupled from the external environment, and that seems to be the signature of collapse, and that is called loss of relational resilience. And look at the mutant. it faithfully reproduces the state after each transition indefinitely. So this gave us the idea of developing a stress test to allow you to predict collapse of biological systems. And then we worked with Ginger Armbrust in the Department of Oceanography and applied this to diatoms. And the objective there was to see how ocean acidification resulting from climate change might alter the resilience of diatoms. And we took this organism called Thalassiosira pseudonana and we put it in this environment where we required it to go across 4 states: daytime state, nighttime state, high nutrient state, low nutrient state. And then we observed it in current day conditions and conditions simulated from 100 years from now. And then we asked what would be the resilience of the organism in the 2 circumstances. And that showed that the organism is going to become more resilient in the future because it is able to have excess energy. When you have high CO2 levels, this organism does not need to concentrate carbon dioxide to fix it. And so that frees up some energy. It can use that energy towards these transitions and dealing with stress. And as a result, it is able to be better prepared for future changes, which might be good for the organism but is not good for the ecosystem. Yeah.
Real quick on the previous slide, so you're sort of messing with the sulfate levels, which the wild type is trying to regulate, right? So presumably there's a reason that the wild type is trying to regulate that.
Yeah.
So is the collapse because the fluctuations are much larger than they would be?
Too frequent.
They're too frequent.
They're too frequent. So regulation makes sense when you have an environment that is changing at a certain frequency. That is, so there's a trade-off, right? So having all the regulatory system consumes energy. You have to replicate it and you have to express it and so on. The amount of energy devoted towards that should be justified in context of how frequently the environment changes. If the environment changes too frequently, it doesn't make sense to regulate. Why not just constantly express at a low level? Yeah. So let's shift gears. So in the last example, the perspective that I wanted you to get out of that is the notion of how the external environmental changes are coupled with internal states and how we can use that as a quantitative metric that predicts resilience, right? And we can get down to mechanistic detail of why these systems are able to deal with changes. Now, this is a very different example. The question here is quite different. It has to do with antimicrobial resistance and tuberculosis, and this is a huge growing threat where the projection is by 2050 there will be about 10 million deaths that will be because of antimicrobial resistance, and 1 out of 4 deaths will be because of tuberculosis. And the reason for that is the scale at which it has infected the human population. The estimates are about 1/3 of the human population has some latent form of the infection, and that you have about 10 million new cases every year and 1.8 million deaths. I mean, if numbers can numb you after some time, but 1.8 million deaths is 3 cities the size of Seattle wiped out in 1 year, every year. So this is the current status, 1.8 million deaths. So by 2050 it's going to get far worse. And we've had this linear thinking about how to kill these bugs. So we think there's a vulnerable point in or gene in the organism. If we target it, then we can kill the organism. But there's a lot of, the organism has evolved in an environment where something or the other has tried to kill it. So it has evolved all these processes to deal with these catastrophic lethal events and is able to circumvent it and figure out ways to resist it. And so we need to understand what are those processes, and that requires a systems approach. And the goal would be to find a way to kill the drug effectively with small amounts of drug in a short period of time. Okay? Because you could keep increasing the drug concentration, but the side effects will also proportionally increase. It's not realistic. How does one get at this? And there are some concepts that have to be appreciated to understand how does one go about understanding this problem. One is what is resistance? Resistance is when a strain requires much larger amounts of drug to kill it, right? So that's genetically based. So you have a mutation in the target gene as well as one example where you can resist killing. Now there are other mechanisms that are not genetically based. One is called tolerance, where it takes longer to kill the organism. The overall concentration of drug required to kill is the same. It just takes a much longer period of time. And there are different mechanisms underlying this, and not many mechanisms have been figured out. The third is what is called persistence, where there is a large population of bugs or cells of that pathogen, and it hedges its bets on a small subpopulation to be in a different state that is tolerant to the drug. That's called a persister-like state. It's typically a dormant state that's not actively dividing. So now you have these two mechanisms playing with, with interesting interplay that underlies resistance, so that you have longer period of killing, even for a fraction of a percent of a population, that can then get a mutation that is resistant and then outgrow the population. So the challenge here is figuring out, we have to look at the scales here. These cells tend to exist as a very small fraction of the overall population. The way people have studied it historically has been to use a drug to kill off all the susceptible cells and then look at the persister cells. Now the question always has been, did the killing effect drive the formation of persisters, or did they pre-exist? And so we developed a technology, I should say Vivek Srinivas developed this technology to find these small fraction of persister cells, and he exploited this property of these cells called translational dormancy. So remember the central dogma, you go from DNA to RNA to protein, DNA to RNA is transcription, RNA to protein is translation. These persister cells are known to be translationally dormant, meaning they can make RNA, but they cannot translate it to protein. And so what Vivek did was he said, I'll develop this technology to exploit that inability to translate. So he overexpressed this fluorescent reporter, so the cells that would transcribe this reporter but not fluoresce would be the ones that would be persisters. And these are the cells, and he was able to distinguish them from these dead cells and also the actively growing cells that are translationally active. And he used a lot of controls, and I'll skip over that. And what he showed was, if you take this population here, that's the population in blue here, that is 4-fold or higher tolerant to drugs, frontline drugs like isoniazid, rifampicin, and so on. And the sobering thing about this was, if you take these populations and you grow them out, you get the original population structure. So you can take the persister cells, put them in broth, they'll grow back up, and they'll look just like the original population. So it's a inherited property, but it's not, these cells are not genetically different from these cells. But an interesting thing happens when the pathogen infects the host. It encounters cues. Remember anticipatory behavior, as I had mentioned early on. They sense things like nutrient starvation, and the life cycle of MTB has been to infect the host, spread, infect another host cell. So every time it infects, it goes into this nutrient shock. So we asked what happens to the population structure when there's nutrient starvation. What you find is that the population that was actively growing with short lag phase, meaning you put it in culture, it immediately grows, the population shifts to being majority cells that take much longer to initiate growth. So nearly 100% of the cells take on this persister-like phenotype. So even before the fight begins, the organism has already put up its shield. So when you put the antibiotic, the organism is in a state that is drug tolerant. Okay, the antibiotic hasn't even come into picture, and you can see that. So the number of persister cells have gone up, and the tolerance to drug has also gone up proportionally. But you treat with the drug itself, you don't see an increase in the persister population. So this is anticipatory behavior. So when it infects the host, it uses nutrient starvation as a cue to get ready for future immune attack or antibiotic attack. And by using the proportion of cells in active growth conditions and nutrient starvation conditions, we came up with this model of switching between actively growing cells that are drug susceptible and these persister dormant cells which are drug tolerant. And we showed that when there's nutrient starvation, the probability of switching shifts and the population becomes more persister-like and highly drug tolerant. And then we sequenced individual cells of these populations, and we figured out what was the internal transcriptome states and what were the mechanisms. And so we built this network, regulatory network of TB, And using the methods I described earlier, I won't go into more detail there. And by mapping the transcriptome onto that network, we were able to figure out what are the active mechanisms that are responsible. And we found there are these stochastic mechanisms at play, which take TB from an active growing form, which has this physiologic state, and through one of several different mechanisms. And TB has at least 70 to 100 different kinds of mechanisms that can drive it to a persister-like state. And the mechanisms are stochastically turned on. So you have this large combinatorial space that allows it to jump back and forth between active growth and persister-like. So which mechanism do you target? What we found is it all converges to this low metabolism state, and we can target that convergent state by using some molecule like cysteine. You can't use cysteine, it's metabolized, but some molecule that can trigger metabolism and shift it to high oxygen metabolism. And we showed when you do that, you can increase the killing effect. So in nutrient starvation, you see the antibiotic has no killing effect, but you add cysteine, and by several orders of magnitude, you potentiate killing. So you convert the persisters into actively growing cells. So that's one mechanism. Now there's another mechanism which is drug-induced tolerance. So once the antibiotic sees the drug, it can elicit active mechanisms to tolerate killing by the drug, which is different from the persistent mechanism is a bet hedging mechanism where cells are switching states. Here what Eliza did was she took this drug that was approved, the first drug to be approved in 40 years by FDA, called bedaquiline, has a delayed killing effect. She took the drug and asked, can you figure out the networks that are turned on when the pathogen sees the drug? So she exposed the pathogen to the drug, collected the RNA from the drug, from the pathogen over time, and then used the network of TB, and she figured out that, I'm going to skip this, that there was a subnetwork that was triggered when the pathogen saw the drug. Okay, the subnetwork had about 70 genes, and we then dove into that network and we looked at who was controlling the network, and we found something really interesting, that this is a bedaquiline kills TB. It poisons the ATP synthase enzyme, which makes energy, and as a result it triggers this nutritional stress-like state, and that then activates this network that is controlled by these two regulators. And we showed if you remove these regulators, you potentiate killing by bedaquiline. And then we found a molecule that can simulate the knockout effect, and we showed you can combine pretomanid with bedaquiline, and that gets a synergistic effect in shortening treatment regimen. Okay. And the last thing that the bug can do is, when it gains resistance, is go to a completely different state where the drug is no longer active. And there's a lot of frontline drugs, and there's in TB you have multi-drug resistance, you have extreme drug resistance, now you have complete drug resistance. So we're running out of these antibiotics, and one thing we have to figure out is, can we reverse the resistant state? So Mario has done this work on E. coli, where he took tetracycline, resistant strains of E. coli, and we have yet to apply this to Mycobacterium. But what he saw was when E. coli gains resistance to tetracycline, its whole network gets rewired, and when it sees the drug, it elicits a very different kind of response. And he figured out what network is controlling their response, and he found out that when the drug is added, the resistant strain turns on efflux pumps to pump out the drug, and to maintain that property, that function, it turns on this regulator, one of these regulators, that shifts metabolism to what is called overflow metabolism. It's a new way of making energy. You can think of that as fermentation in presence of oxygen. Okay, because the efflux pump has occupied all the real estate on the cell, so your oxidative metabolism, the respiration systems have got no space to operate. So now it has to operate in a pseudo anaerobic condition. That's called overflow metabolism. That supports drug resistance. So his hypothesis was, if we disrupt this network, you should reverse tetracycline resistance. Skip that, and that's what he showed, is if you remove the regulator, the resistant strain which cannot be killed, what we're looking at here is the lag phase. So tetracycline is a bacteriostatic, so it inhibits growth. So this is wild type. it is unable to grow. It takes, in high doses of tetracycline, it has this infinite lag phase. The resistance strain has a short lag phase. You remove RK and it can shift it. So you can reverse resistance. So now we looked at different phenomena. At each level, we're looking at different kinetics, different mechanisms, some operating at single cell level, some induced by adaptive prediction by sensing host cues, and then you have these resistance mechanisms. And so you can think of a number of different ways by using these mechanisms interconnected of how you can now mix and match drugs that have synergistic effects. And I'll skip this, but the basic idea is instead of just focusing on this one drug that kills the organism, you think of how you disrupt tolerant systems, how you disrupt persistent systems, how you disrupt host-induced expansion of tolerance, and so on, to come up with drug cocktails that are much more inspired by the systems biology of the organism rather than reductionist understanding of which gene is essential. It goes beyond essentiality. And so this is the last vignette, and I'll go through this one a little quickly because of time. And here that the thing we're addressing is the question of the growing burden of healthcare. As you know, it's becoming more and more expensive to get drugs that are effective. And there's a paradigm now that the poor responsiveness has to do with differences among individuals. So every individual is unique. And there's a map that, given a certain mutation in an individual's cancer, I'm using cancer as an example, there's a drug that they're going to be most responsive to. So we have this linear kind of understanding of mutations to drug. And just as we were wrong with this one gene to one disease paradigm, we're wrong here as well. Because in reality, when you sequence tumors, you have thousands of mutations. The mutations interact with nonlinear dynamics. And oftentimes, even if you have the mutation that you think a patient that should make a patient responsive, because of some nonlinear interaction, then they're not responsive to that drug. And so this heterogeneity across patients, patient tumors especially, is responsible for poor responsiveness. So even when you find a drug that seems to be really effective, across a population, there's only a fraction of the patients who actually respond to the drug. And this directly translates to the drug discovery pipeline, where we start with tens of millions of compounds. In brute force, we then identify that one molecule through a series of preclinical trials and phase 1, phase 2, phase 3. And by the time you find a drug, you spend $2.5 billion on finding that drug. And even then, you're ineffective. You're essentially treating a very small fraction of the population. So our question was, can we uncover this map of mutations to phenotype? Okay. And the way we've done that is, I've described this already in the first part of how we uncover networks. Here's a little more information on that. So I've described how we build a transcription factor DNA map, the RNA target map, and so on. So there's a large knowledge base that has been created through decades of research. So we take all of that information, we combine that with public data sets such as the Cancer Genome Atlas, which are described as this database where tens of thousands of tumors have been profiled from as many patients. We combine that through the signal pipeline of finding the networks that are perturbed across the patient cohorts, and then we build this map that I mentioned in the early part, which gives you a map of mutations and how they causally alter the networks that control these gene modules associated with phenotypes, disease phenotypes. And this mutation to phenotype map is for the entire patient cohort that you've studied. What we've then done is develop methods to then map individual patients to figure out what is unique about a given patient. Because a given patient does not have all the dysfunctional networks, they have a subset of those networks in some unique combination. And then based on that, we can stratify the patient, predict their risk of clinical outcomes and responsiveness to therapies. We can accelerate drug discovery. Rather than treating all patients as a random average population, we are able to match the drugs to patients who have characteristics that predict that they would be responsive. And in that way, we can select therapies based on patient characteristics and predict who's going to respond and who's not going to respond. And we've done this for both solid tumors and for liquid tumors with glioblastoma with a number of different collaborators and partners. And for both cancers, we built this map. And there's different sets of mutations that alter networks in brain cancers, different set of mutations that alter networks in blood cancers. And then now we can use these networks to stratify patients. So as an example, for multiple myeloma, we can now take pre-treatment profiles. So when patients come in and are profiled, even before they go to therapy, we can then predict which of the patients are going to have low risk and which patients have high risk, which patients are more likely to respond to therapy or combination therapy. And so we can risk stratify the patients, and the score with which we can, this is a log scale, with the network based approach is orders of magnitude better than the current best in the clinic right now. But we can go for, there's a question back there. Yeah, so you can look at which function the mutation maps to, and in terms of the gene, the consequence of the mutation, is it non-synonymous, or and whether that gene is involved in a pathway that is known to be associated with some phenotype, right? It's not complete, but iteratively over time you can begin to then find additional mutations that have functional consequences. But you do far better than just going with one or few mutations. You can look at all the mutations that were previously unseen. Okay. And what we've done in collaboration with Mike Jensen at Seattle Children's and with Charles Cobbs up in Swedish, is we've shown that if you find mutations, that this method can find mutations that alter the tumor microenvironment. And here's one example. So there are mutations in certain genes that alter the network that controls antigen processing and presentation. So we predicted that patients who have these mutated networks would have increased tumor lymphocyte infiltration, and that's something you can go and test. And further, because we know this network, we can then predict other molecules that can potentiate the tumor microenvironment by altering the process, the downstream process. And so you can combine the molecule with, say, CAR T cell therapy to potentiate immunotherapy. And here's an example where we show that we can rationally find drug in this example, we combine a drug with a microRNA, and this drug is known to be an effective targeted therapy, but it's very toxic. And we showed by combining with the microRNA, you can lower the dosage and have the same effect on proliferation. So I've ran through this, but if you have interest in learning more, and this paper kind of documents extensive analysis. So we've even done genome-wide CRISPR-Cas9 screens with Patrick Patterson at the Hutch to show that the network has identified a lot of disease-relevant networks that control proliferation. We've also shown targeted siRNA, microRNA mimics drugs individually and in combination, and so forth. Okay. So the basic idea I wanted to get across from these vignettes is, although these questions seem very disparate, from host pathogen interactions to microbial ecology to cancers, the basis of how cells respond to environmental change, how cells interact with each other, and how they evolve to environmental change has some